So for this experiment, we'll be doing a group experiment performed by a group of 4, and these will also be your writing groups. So we're going to create artificial foraging patches, or we're going to use a food source on an index card. In our case, we'll be using soaked cotton balls with a sugar solution. We're going to count and observe the number of ants that recruit to the food patch over time. After an hour, count the number of ants on each patch, and note whether there are more than just fire ants present. So if there's other types of ants, other insects, just note that. And then you're also going to say, you don't need to know the species itself, but you can say like small black ants, fire ants, or large black ants, other descriptive words. And at the end, you're going to put some gloves on, and you're going to pick up your index card and place them into a uniquely labeled plastic bag with all the ants in it. Our experimental treatments will be height. So you're going to have your control, which is on the ground with a cotton ball on a index card. And you're going to have one that's on a foam block that's 10 centimeters high. Then you're going to have one on 2 foam blocks, because it's 20 centimeters high. So when talking about experimental design, a good experimental design in a manipulative experiment will change only the variables of interest, will manipulate each variable independently, will exclude as many other sources of variation as possible, and will have a control. So in field experiments, where does variation come from? There is natural field variation, which is inherent spatial variation in the environment, temporal variation, so weather, morning, evening times, and there's animal behavior. And then there's experimental variation, which is like data collection error or biases, random events or intrusions, or poor experimental design. When talking about properly designed experiments, it will reduce the potential effect of other variables by trying to control the variation inherent in the environment, and attempt to reduce the sampling bias and variation inherent to experimentation. So in manipulative experiments, add, modify, or remove individual variables or a treatment in an attempt to isolate the cause. So it won't just be a correlation. You'll try to manipulate a treatment to determine the cause. So a treatment is a manipulated variable. The control is unmanipulated variable, and you'll have replicates as well. So repeated experimental units of each treatment in control. For ours, we'll be doing 2 replicates. So you have 6 cotton balls total. So in this example, you see how many treatments there are. There's 1, 2, 3 treatments. How many numbers of replicates per treatment. So you can see that there's 4 flowers per treatment, so that's 4 replicates. And then you can see the overall sample size total. And then there's 2 different types of controls. There's true controls, which usually leaves all variables unchanged. And there's procedural or negative controls, which mimic some aspect of manipulation in order to affect the outcome. So when accounting for variation, that's why we have manipulated controls. Sometimes manipulating organisms themselves can cause unintended effects that we call experimental artifacts. In order to control or deduce what these artifacts are, we add additional control treatments to examine the effects of them. So for instance, you have cages that are meant to exclude organisms, but it also causes shading on the plants. So to ensure your variable of interest is the only variable manipulated, you should ensure that all your treatments and controls experience similar conditions to all other environmental or experimental aspects. This is accomplished by interspersing and randomly distributing your treatments. So in this example here, we have the presence of owls reduced squirrel foraging. So all these blue blocks are where you put your treatments. Some are in open spaces, some are near forests. And then the owls will go to different areas, right? So you can achieve this by randomly deciding where your treatments are placed. So you could take a coin to your side, do heads and tails, or do a random number generator. Just make sure that they're not all placed in the same location or placed like around an ant bed or anything like that. So in this example here, you can see that there's white treatments and there's black treatments. Whenever you disperse your variables, you don't want them in 2 very, very different areas. So you see that the black treatments are along a path, and the white treatments are just in the woods, make sure that you try to keep your all your variables and all your treatments in the same type of area, but just dispersed in that area. So this is site selection. Keep as many variables the same as possible other than the one you are manipulating. So footpaths, trees, forests, just make sure that it's on the same substrate around the same area to not have a biased treatment. So whenever we get there, we'll be taking coordinates. You can find this on your phone. You'll be doing site information, so just where you put your treatments, what type of light it is, what the weather is. And then you also have the ant observation, so anything you observe about the ants or their behavior or any type of ant, it would be listed here. And then here's the required data to take. The number of ant at each patch after an hour. So at 30 minutes, 15 minutes, and 45-minute increments. And then the time of patch placement and the patch pickup. So the time you put it down, the time you pick it up. The number and species of ant. So I had like 15 fire ants and 6 tiny black ants, whatever you get. You need the GPS coordinates of every patch. And then other metrics will be hypothesis dependent, but may include food type, tree girth, human activities in patch range, or other relevant factors. Yeah. So we'll have a data chart like this, and you'll put all your data on this chart.